All right. Good morning. Hopefully you're feeling energized by that extra hour of sleep over the weekend. It is election day, so because of that, we do have a modified schedule. Classes ending at 9:20. If you could all try to help keep me accountable to that 9:20 time, that would be appreciated, because I'm going to forget, I know it. Let's see. I started passing back your content checks. I will continue doing that throughout class today. Otherwise, I went through the online portion of the content checks, and I wanted to just give a demonstration. Yes, my camera is working. All right. Some of you were confused about how DNA gets condensed, right? And I didn't have a visual, but I have a visual this time. So if we look at this bead of strings, right, we can assume that this is the 30 nanometer fibers. This is the DNA wrapped around the nucleosomes in that zigzag fashion, right? Now the radial loop domains seems to be where the confusion was, so I'm going to start looping this, twisting it up so you can see kind of a visual of how this is going to condense. And it takes a little bit to get to that stage. Tried to put it up on the screen so that we all could see it. Oh, there we go. It's starting to condense. Oh. These beads are a little tedious. But you can see it's already starting to bulge out in some places with those loops. Right? So when it's all the way twisted up, it's actually a lot shorter. So that's how those radial loop domains help condense that DNA down. So hopefully this visual kind of helps wrap your brain around it.
It goes for break, right? If you break something, it's going to get shorter.
Yeah, if you break something, it's going to get shorter. All right, the other visual that I didn't have at the time was topoisomerase, helps relieve supercoiling. And I wanted to give you a visual of what supercoiling would look like. So if I try to break this apart, eventually it gets knotted, it gets twisted, it's a mess. I can pull and it's just not going to break apart anymore. All right, so topoisomerase helps prevent this from happening in DNA as you start to unwind it. All right. So hopefully those visuals kind of put something into perspective for you here. I had a question about, there's a lot to remember in DNA replication. Yes, there is, right? So I have made this slide as like a one-slide review, and I included it on your PowerPoint slide. But first, right, the DNA is going to unwind, and that involves 3 proteins, all right? The helicase, the single-strand binding proteins, which is a class of multiple proteins, and the topoisomerase, as shown in this orange box on the side of the diagram here. Right? And then second, the DNA replicates. All right? So that involves RNA making the RNA primer. It involves DNA polymerase 3. And it involves DNA polymerase 1 with the ligase. Right? And again, all those proteins, well, most of those proteins are shown in this purple box. What is not shown here is DNA polymerase 1 in this particular image. Right? DNA polymerase 1 is the one that kind of takes out those RNA nucleotides and replaces it with DNA. Right? And then the biggest difference, right, between the 2 strands is just directionality. Right? So this is going to take some studying. You're going to have to want to review this several times, draw it out. I don't expect you to, like, me teach it to you and then you would automatically know it off the top of your head. All right? It does take some studying, take some time outside of class to learn this. Hopefully this one slide visual can help you out a little bit here. All right, but I actually wanted to start off class by talking about the scientific literacy review. So hopefully you have completed that it was due today. And this scientific literacy review was all about the peer review process that happens in scientific journal, scientific literature. Right? And so my review of this assignment is actually kind of a shorter one today. because all I want to ask is, what are your questions? All right, so question 2 of part 2 asked you, do you have any questions about the peer review process? Right, and now is your time to ask me about it. I have gone through this peer review process. I've had to deal with several aspects of it. So if you have any questions, please let me know.
Do scientific journals tend to have like certain biases on beliefs, or like or only accept journals from specific types of people? Because I feel like it almost, the way it was described made it sound like it was way too easy to filter out and withhold information inside of a science journal.
Yeah, so do they have biases? I would say they like to not have biases, but there's going to be some implicit bias in any journal review. For example, the reviewers, they don't give out their identity, and they should not know who the authors of the paper are to kind of help filter out any biases against specific people. But there are biases, right? If your lab studies one method of doing this process, studying, say, this particular plant, and another lab is doing a whole different process, that process that you do not like, you might have some implicit bias when you're reviewing that paper. Right, so we try not to have biases, but there are. Yeah.
I feel like that would be very likely to happen if 90% of papers get rejected.
It could be, and most of the time papers are rejected simply because they're not put together enough. Like there's not enough for it to be published yet, right? It tells part of the picture, but not the whole picture. And when you publish a scientific article, you want the whole picture given out. You don't want too many loose ends. What other questions do we have?
The assignment talked about how there are critics about the peer review process. Do you know if those critics have any alternative processes?
It's a great question. Yes and no. I know there are journals out there that do not have a peer review process, right? They're not considered academically rigorous because anybody can publish in it. There's nobody checking your work, so you have to be kind of weary about the papers you're reading there. So yes, there's that alternative method. It's not a great alternative method though, so I don't know if there's an academically rigorous alternative to not having peer review. Yeah. Peer review is a long, tedious process. I can see why there's critics. Any other questions? I know you all have them. You had to write down a question in your assignment.
Why is it that even though if the reviewers actually kind of like they give it the go-to, like it's good, but why is it like that the editor has the final say, even though the reviewers who are actually knowledgeable in that area say it's good?
Yeah, why does the editor have the final say? Normally the editor has the final say more or less on does it fit into this issue, not does this journal fit our criteria of being academically rigorous. So a lot of times issues will focus on a particular theme, like maybe it's the plant physiology theme, or maybe next week it's all about plant taxonomy, so naming things, or new species being found. So maybe it doesn't fit into that particular theme, and then that's where the editor might have more of a final say. I think it's really rare for an editor just to outright reject a paper because they don't want it ever. It's more like they don't want it in that particular issue.
So I guess in a sense, I'm thinking of the monthly issues of Reader's Digest. I like to read those. There's actually, I know that some people kind of give them flack, but there's actually a lot of good stuff. They do take very, like, they do put a lot of research into the stories they provide and the information that they give out.
So other magazines can have rigorous journals.
A lot more open source though.
Yes, yes. Sarah, you had a question?
Yeah, how long does like this process take?
Oh, that's a great question. It could be relatively quick, and when I say quick, it's like 6 months. It could be a couple of years. It could take a long time. Yes, because sometimes you'll submit your paper, it takes a while for it to be reviewed, you'll get back comments on your paper. Then you'll address those comments, and they give you a couple of months to do that. You'll submit it back to those reviewers. They might come back with even more comments that you have to address. So this process can drag on for like a year or two in some cases. Yeah.
Actually, having to write something like that on computers, that would be a nightmare.
Any other questions about the peer review process? Yeah.
Free from pop culture, that would also be awful.
So Sarah, you had a question?
Yeah, how is, oh, sorry, my series going on. Oh, sorry. How is it the feedback can be given to the author from the referees in person, on paper, or email?
It's all on paper. So you'll get like, typically it's like an email back with like a packet of, this is reviewer 1's comments, this is reviewer 2 comments, and reviewer 3 comments. They typically write their comments. Depends on the journal, they might have a separate document, they might honestly just like annotate the paper itself in some places.
Yeah. You get paid to do that.
They don't get paid.
What?
Reviewers are typically volunteers. Yeah, volunteers in your field that want to make sure that we're upholding this like academic need, making sure what gets published is rigorous enough. Yeah. In fact, a lot of times editors can be volunteer positions too. Depends on the journal. Yeah. All right. Anything else? Alrighty, it looks like, oh, my slideshow is still up. It didn't time out. All right, well, there's no other questions about peer review. We're going to head into content then. So we started talking about why cells might divide last lecture. We just briefly got into it. So now this week, we're going to learn about mitosis and meiosis. Maybe you've already learned about this. Maybe you need a refresher. Maybe this is the first time you're kind of diving this deep into it. All right, so let's talk about kind of like the precursor to a cell dividing, right? Why does a cell divide? So cell division, right? Cells do not just appear out of nowhere. They arise through a pre-existing cell, right? So you have to have a cell in order for another cell to appear, right? Cells don't just spontaneously appear. And there are two types of cell division. We can have mitosis and meiosis. Right? Mitosis is going to create an identical cell, whereas meiosis is going to create a genetically unique cell. Right? So in humans, right, and I'm going to talk about cell division mostly in humans here or mostly in mammals. Right? In humans, we're going from a diploid cell to an identical diploid cell. So remember when I say diploid, diploid means we have 2 chromosomes per pair or per set that are homologous to each other. Whereas meiosis, we're going to go from a diploid cell, so having 2 pairs per set, to a haploid cell, which is only 1 chromosome per set. Right? No matter what type of cell division we're going through, whether it's mitosis or meiosis, it's always going to be accompanied by this process called cytokinesis, which is the splitting of the cytoplasm of the cell, right? Which is kind of what's shown in this image here. You can see the cell is physically splitting into 2, all right? So that would be cytokinesis. Now we're going to go over all the steps of mitosis, meiosis, and even talk about cytokinesis in a little bit more detail, all right? But this is like my broad overview slide of what is going on. All right, so let's look at meiosis and mitosis in a little bit more detail, because again, that's going to be the focus of these next, this lecture and Thursday's lecture here. Meiosis, right, we go from a diploid to a haploid cell, is going to lead to the production of gametes, right? So if we're talking most organisms out there, gametes will be called egg and sperm, right? The key here is that these gametes, these daughter cells, have half the amount of the genetic material as the parent cell. Right? We've divided it by 2 because we've gone from a diploid cell to a haploid cell. All right, so meiosis leads to the production of gametes. Mitosis leads to the production of what we call somatic cells, or basically every other cell in our body that are not gametes. Right? This is where genetic material is directly copied. Right? The daughter cells should be identical to the parent. We're going from a diploid cell to an identical diploid cell. All right, so overview again. Meiosis leads to gametes, which are haploid, contain half the amount of the genetic material. Mitosis leads to somatic cells, which are all the other cells, and are genetically identical to the parent. All right, I'm trying not to go too fast. All right, so we're going to look at mitosis first. All right, mitosis is where we're getting that genetically identical daughter cell. Okay? So, why might a cell undergo mitosis? Well, there's a variety of reasons. If you're a single-celled organism, this is the way that you reproduce. Right? You go through asexual reproduction. Right? So you're simply dividing yourself, pulling yourself into two to create another individual.
I read somewhere that cells can have that cells can have sex, but it's just not like that. Like they can merge each other. They can merge, and then it's just one cell with both of the original cells' DNA together.
That is true in some cases. Yeah. So in fungi, that's really common. Fungi can do that. Bacteria have a way of doing this where they can exchange genetic material between one another. Yeah. We're not going to get into the details of that type of exchange of genetic material.
It does provide more genetic diversity, but it's not technically reproduction because technically you end up with less.
Yeah, so it does increase genetic diversity. And we'll see those words genetic diversity throughout the entire lecture here because genetic diversity is a very important thing to have. All right, so if you're a single-celled organism, you'll go through asexual reproduction. If you're a multicellular organism, you might go through mitosis for a variety of reasons. It might also be asexual reproduction. So plants, if you've ever seen a plant that you can take a little piece of the plant, stick it in soil, and then it grows up into a new plant, right? That's a form of asexual reproduction, right? There's no merging of gametes involved. It's just mitosis all the way. But also organism growth, right? So think about the number of cells you have in your body today versus when you were a toddler, right? You had to increase the number of cells, so you're growing. It might be repair of organisms, right? Maybe you broke a bone, you fell and scraped your knee. You need to repair something within your body, right? That is also going to be mitosis here. All right, so knowing what you know, and we haven't done too much yet, but you should be able to answer this review question. Which of these diagrams is mitosis and which is meiosis? Give it a try. Yeah. Yeah. I'm going to give you still 2 minutes, but seems like you got it.
I missed what was on the slide. What is the production of all other cell types is referred to as what?
Somatic cells.
Somatic cells.
Yeah.
Yes.
Their sisters, but they're not twins.
Before they're just floating around. Before they realize.
Yo. Dude, that is.
And they're like,
And I think we're starting now. And I think it's that.
wait, why is this happening?
Well,
I do not have an answer for you. That one, yeah.
those are only two. I don't know why it's right now. This is
Yeah, I'll take it.
one of So they split here once, and then they split here again. Yeah. Yeah. Yeah.
That's why there's a restriction on
Yeah, and I'm pretty sure they like swap or exchange something which causes the variation, but also can cause
Maybe it's just the-
stress later on with the family.
It's a bunch of variation, like being like a puzzle, like being rearranged.
Yeah, and we'll talk about that when we talk about meiosis, but that's, it's called crossing over, or genetic material is exchanged to create genetic material. Yeah. Okay, she's a senior. Okay, I saw her little slide up in the hallway, outside of the bio office. Yeah, for all the seniors, we get like a slide and post it. And I'm like, I recognize that last name.
Guess who?
Yeah. All right. So if we're gonna look at these two diagrams, which one on my right or my left is going to be mitosis? It is going to be the one on the left. What led you to that conclusion?
Just identical.
It's identical, right? So the parent cell looks identical to the daughter cells, right? You could also say that our ploidy level does not change. We're going from a diploid to a diploid cell. Whereas if we look on the right, right, the daughter cells do not look identical to the parent cells, right? This is meiosis, and we'll talk about meiosis more on Thursday. All right, so let's talk about cell division. Why does a cell divide, right? What leads it to go through this process? Well, we can't talk about cell division without talking about the cell cycle. So the cell cycle is a series of events within the cell that will ultimately lead to cell division, right? It's a highly regulated series of events as well. Think of it as kind of like this is the cell's lifespan, right, or its life history, and it's going through its life history, right? Now there are several different spots or several different steps in the cell cycle, and we'll go over them and what they each mean, right? But notice that the cell is kind of its life history, but it could repeat its life history. So it could decide to divide once, right? And then it's not going to die. It might stay alive, and then it could repeat twice, 3 times, however many it's predetermined to divide.
Or if its DNA is really screwed up, is live forever.
Potentially. I know jellyfish, their cells might die, but their organism is technically something that lives forever. That's kind of a cool
Until it
little
gets eaten.
fact.
Yeah, I do know about that. The moment it's eaten, it doesn't survive that.
Yes. All right, so interphase is what we're going to talk about first. Interphase contains the first 3 stages of the cell cycle. Right, so if we look back at the cell cycle, we see there's this G1 phase, there's an S phase, and there's a G2 phase. Right? Combined, all 3 of these phases together, we call interphase. Right? So if we look at the breakdown of these different steps, right? G1, it's called the first gap phase, which is why it's called G1. Right? This is where the cell is growing. Right? It's starting to prepare to divide. It's growing in size and might start replicating certain proteins it might need for cell division. Right? So G1 is growth. The S phase, this is where DNA replication is happening. Right? So we know how DNA is replicated. Right? It's the S phase of the cell cycle where this replication actually takes place.
That long?
And the entire replica genome is duplicated, yes, and in humans it takes 10 to 12 hours, which, fun fact, right now you have some good trivia under your belt. After the S phase comes the G2 phase, which just like G1 is called the second gap, right, so G2. And this is involves more growth of the cell. And then this is definitely where all the proteins that might need to be duplicated, might need to be synthesized in order for cell division to occur, happens. All right, so interphase, we get growth, we get DNA replication, we get more growth, right? Basically, the cell is prepping itself to divide in interphase, right? Now, I like this image of interphase here, right, because it's showing that the DNA inside of our nucleus, right, isn't super condensed. It's still it's condensed, but not super condensed, right? So it kind of looks like a plate of spaghetti at this point. If you were to look at it, you would not be able to pick out a homologous pair, right? It's pretty complicated, pretty convoluted. All right, so we have interphase. And I forgot I had an extra little image here. We have interphase, so that's G1. That's the S phase. So when we're looking at drawings of chromosomes, this is where we go from a singular chromatid to having sister chromatids, right? And then we have more growth in that second gap phase. All right, so collectively, the first 3 steps are known as interphase. Do I pop up there? Yes, it pops up, right? After interphase, after the cell has prepped itself to divide, right, then we actually get the division happening, right? And that is the M phase, right? That is where we go through mitosis and cytokinesis. Now, there could be an instance where this cell is not going to divide, or it's postponing cell division. And that's where we get this extra little loop in the cell cycle that we call the G0 stage, right? So if we're looking at our diagram here, G0 stage is kind of like this offshoot of the G1 stage. If a cell is not going to divide, or it's postponing division, it's waiting for the right signals in order to tell that cell that it needs to divide, it's going to indefinitely end up in this G0 loop, right? And just kind of hang out there, right? It's not going to grow, it's not going to synthesize more proteins, it's just hanging out.
What would cause a cell to get in that G0?
What would cause a cell? Well, we know it's not going to divide, so maybe it's part of an area of your body that doesn't currently need any repair, or maybe the cells are healthy, we don't need to replace anything. It's hard to say what would keep it there. It's easier to say what would knock it out of G0, because a cell is typically not going to divide unless it gets an external signal that it needs to divide. We could think of neurons. They don't go through mitosis or meiosis. They're always going to be in G0 because they don't divide. Yeah, it's a good question, and I'm sure there's a variety of other answers out there as well.
And so when I was referring to the idea of cells dividing indefinitely, how does the process of cancer happen?
So cancers happen. I'm going to go to my next slide here, and actually it'll be the slide after, when certain checkpoints in this cell cycle do not happen, right? So I'm going to leave that open-ended for now, and we will get to it. So
Some cancers are genetic.
there are some that are genetic. Yeah, you're more genetically predisposed. So if a cell is deciding to divide, it has some different factors, right? It could be an external factor. Environmental conditions, right? You fell, you scraped your knee, you need to replace those skin cells that have been lost, right? So the environmental conditions outside of the cell. It could be different signaling molecules saying that we need more cells in this area. So think of like hormones, like growth hormones, right? It can also be internal factors, right? Inside the cell itself, right? So we have cell cycle control molecules that control the cell cycle, control whether or not the cell can move on from the G1 phase to the S phase to the G2 phase. And there's also certain checkpoints that the cell has to pass in order for it to move on to the next stage. Right? So there's a variety of reasons of why a cell may or may not divide. And it kind of depends on the situation of the cell itself, whether it's external need or it's a need within the cell itself. All right, so we have these internal factors, right? These cell cycle control molecules and these checkpoints. I want to briefly talk about these checkpoints because they play a really important role in our cells. All right, so within our cell cycle, there are these regulatory points where a cell is checked to make sure everything is proper before it can move on to the next phase. Right? So in eukaryotes, right? So not prokaryote, in eukaryotes, like ourselves, like mammals, like insects, like plants, there are 3 checkpoints. Okay? One checkpoint is in the G1 stage, one checkpoint is in the G2 stage, and one checkpoint is in the metaphase stage, which is part of the M phase. Right? So these checkpoints, right, and we're not going to go into the checkpoints. Your textbook might go into more detail about this, but we just don't have time, right? These checkpoints act as like a sensor, right, or like a security gate. If you don't have the proper credentials, it's not going to let you through. Or if something has not properly occurred within the cell at that point, it's not going to let you through, right? So for example, in this image here, you can kind of see some of the things that these checkpoints are looking for. So the metaphase checkpoint is going to look for chromosome spindle attachment. We haven't talked about that yet. This will make more sense in a few slides. The G1 checkpoint is going to make sure that there's enough nutrients, right? That enough growth has occurred or there's potential for growth, and to make sure the DNA is not damaged, because we don't want a cell to divide if the DNA is damaged. Right? So this gets to the point of cancers, right? There are some cancers that can get through these checkpoints with damaged DNA that might cause cancerous growth, right? When these checkpoints are not working properly, right? So we want these checkpoints to work properly. And then the G2 phase is going to check for cell size, right? Is it big enough that it could actually divide, right? Is there enough substance to the cell? And to make sure that the DNA has replicated properly. Right? And to make sure it has replicated. So again, there's these security checkpoints that the cell has to go through in order for it to complete the cell cycle, to complete cell division. Right? If problems are found, the cell can either fix these problems, right? Or the cell might end up dying because it can't fix that problem and it can't go through the checkpoints. Right? And like I said, loss of these checkpoints, that's what leads to mutations and some cancers. Right, so these checkpoints, super, super important. I'm actually going to hold off my questions for a bit. All right, so I want us to do a group review. Right, what are the 5 stages of the cell cycle and what happens in each stage? And I'll give you a couple of minutes. Lucy, you had a question?
Medication that permanently changed the genetic makeup of my genetics. So I had to take a lot. That was the one I had to take lots of facts for.
Okay.
But I have a genetic disorder. I had a genetic disorder where when my body produces acne, it doesn't, like, they don't show up in these tiny little ones. It's always like these really large ones, and like it doesn't like it has a really hard time like undoing that. I know where it comes from. It comes from my dad.
Okay.
And it was interesting because I took a pill, and so now my genetic makeup didn't permanently change so that it doesn't happen anymore.
I did not know that was a genetic thing. I wasn't actually at that. Yeah, that's interesting.
I think it's different than the one my dad had.
Yeah.
Because I believe my dad used to take vitamin medication, but mine was more of a recent one. I know they had to like constantly check my blood pressure just to see if I was pregnant, even though I was like 12.
Yeah.
Just like I'm not sure.
Abundance.
I know, because it was like guaranteed birth defects.
Yeah. It's not a great medication to be on. And it does wonders, but it has a lot of side effects.
Sometimes they say it's like, you might have to take this medication again, and it's like, oh god, I don't want to go through that process again.
Mitosis. They're going to be different. So the cell's gonna start diploid, it's gonna end diploid. Yes, because we wanna create something that's like dental. Now this is, we're talking about like mostly humans. If the cell started out diploid, it's gonna end up triploid. It's still gonna be identical. Yes.
It's like a loop that tells.
There is an instance where if you have triploid genes, I forget which set. Yeah, something has happened, damage. That's what leads to genetically different cells.
Look so close. Yo, click it.
Crossing over occurs in mitosis, and that's what leads trying to explain this is what we've already learned. This is chemical and terminology. The one that I was the short time frame. I don't think
I think that's it.
So it essentially has to go through that's why you're allowed
Doesn't he like do
get to that point?
Yeah.
You stop me about 5 minutes before we're done.
When is that? Is this like at least it's like,
Yeah.
oh, I think just want to go keep like this yeah.
No, like you do it, yeah.
You have to do it once
Yeah, you have to have a sleep. Yeah, that's actually a common issue.
But I don't want to like change it. So everybody's going to change it.
It should work. That's the way it's worked in the past. So with this website redesign, maybe that's something that got lost. I don't know.
But I'll just tell you when I do it.
Perfect, yeah. Yes.
It's just next step 5. Why is it like DNA only contains 5
You have to wait.
Coded wrong, I think.
Dude, found it.
Saying that this question is created, forgot to be better, I'll have to go back and manually grade it, I think. All right. So what are the 5 stages of our cell cycle? What is the first stage? It could be the M phase. We're going to start with the G1 phase. Yes. What comes after G1 phase? S phase. What happens in the S phase? DNA synthesis. What happens in the next phase? More growth, and that's what phase? G2. And then after the G2 phase? M phase, and that's where the cell divides. Yes. What is the phase that the cell will be in if it doesn't divide? G0. Yes. All right. So we kind of glossed over the M phase. We are now going to focus on the M phase, because the M phase is where mitosis occurs. So within the cell cycle, we are now in the M phase, which means our DNA has been replicated, right? And our cell is ready to divide. So the M phase is where cell division is actually going to occur. Now, we're talking about mitosis here, right? So the cells are going to produce two new cells that are genetically identical, right? They should be genetically identical to the parent. Now, when we talk about mitosis, we might say that there's the parent cell or the mother cell, right? And that the resulting cells that happen because of mitosis we would call the daughter cells. Right? So in order to divide, we're going to go through mitosis, which includes all the phases of mitosis: prophase, prometaphase, metaphase, anaphase, telophase. And at the end, we're going to have cytokinesis, where the cell actually splits and divides its cytoplasm. Right? Now we're going to go over each of these steps in detail here. Right? So mitosis is something that if I tell you this is the ploidy level of a cell, right? This cell is going through, say, anaphase. I want you to draw this out and tell me what is happening. Right? So on an exam, you should know these phases. You should know the order of these phases. Now, there are some terms or sayings that help you remember the order of this prophase, prometaphase, metaphase, anaphase, telophase. Right? I have one where I think about if you just adopted a new puppy who is not potty trained yet, you might have to use a lot of PP maths. Right? That's how I keep it in my mind. But I know there's some like a sentence phrase that you can repeat that'll help you remember the order here. I don't know those sentence phrases off the top of my head. I'm sure a quick Google search would show you a bunch of them. All right, so the M phase. We're going to start talking about these specific phases within the M phase. And we're going to start with prophase. Right? So in mitosis, we have prophase. What is happening in prophase is that the chromosomes, which have already been duplicated during the S phase, are now going to be joined as sister chromatids and become condensed. So it's no longer going to look like a plate of spaghetti. Now these chromosomes are going to be condensed. We will be able to see the individual chromosomes quite clearly at this point. Right? So I'm going to draw this as we talk about it, because I think drawing it out could help a little bit here. I'm going to have my cell. Right? I'm going to draw, let's do blue and red, because that's what I have on this side here. I'm going to draw two homologous pairs. I think that's going to be the easiest way to do it here. All right, so my color in my image is representing where the genetic material came from, either mom or dad, and the sizes are representing homologous pairs. So the two small ones are homologous to each other, and the two big ones are homologous to each other. Now I've drawn them as these X's, right, because they've been condensed, and because we've already gone through replication. All right. So there's more that happens here, right? The nuclear membrane is going to start to disappear, right? So the membrane of the nucleus is going to start to disintegrate, right? So I'm going to draw that as dashed lines. So remember, this is all occurring in the nucleus. Right, but during my prophase of mitosis, that nuclear envelope starts to disappear. We also have this mitotic spindle starting to form. Right, this mitotic spindle is going to play a role in pulling our chromosomes apart. And we'll look at it on the next slide here. But I'm going to draw this mitotic spindle as starting to occur. All right. That would be prophase. So, chromosomes have been duplicated. They are now condensed. the nuclear envelope starts to disappear, and the mitotic spindle starts to be formed. Now, what is that mitotic spindle? All right. This mitotic spindle plays a huge role in organizing the chromosomes during cell division. Right? So it's composed of microtubules. Right? So that should be a familiar word. There's microtubules in the cytoskeleton of the cell. So it's composed of really long, thin protein filaments, microtubules, and centrosomes, which is where the microtubules originate out of. All right, so this yellow part here would be our centrosome, and our microtubules would be all these green kind of stringy bits that are coming out of our centrosome, right? Long protein filaments. So the centrosomes themselves have actually already duplicated. They are ready to go when we get to prophase. All right. We could also call them microtubule organizing centers. Right? I think that's pretty wordy compared to the word centrosome. They have been duplicated during interphase when DNA of replication has occurred. So there's 2 of them when we get to prophase. And each of these centrosomes are going to define a pole of the cell. All right, so if you think about planet Earth, we have 2 poles. We have the North Pole, we have the South Pole. Opposite ends of the planet. So when we're talking about the poles of the cell, we're talking about opposite sides of the cell. So eventually we'll see these centrosomes be at opposite sides of this cell. All right, so in prophase, the mitotic spindle has just started to form. So when I've drawn it on my drawing, I've drawn a dot representing my centrosome, and little tiny microtubules starting to come out. Now they're not at the poles yet, but they will migrate there. All right. So prophase with our mitotic spindle starting to form. Really try not to go fast through mitosis. Let's look at the next phase, which is prometaphase. Now, some textbooks, particularly if they're an older textbook will not show prometaphase as a stage. But we are going to consider prometaphase as a stage. In most modern textbooks, the way we think about it nowadays, we consider prometaphase an official stage. All right, so during prometaphase, again, I'm going to draw my cell here. My nuclear envelope is completely gone at this point. All right, so I'm not even going to draw any dashed lines. I don't need to because my nuclear envelope is gone. All right, my mitotic spindle with my centrosomes. They're now going to be at opposite sides of the cell. So I'm going to draw 2 centrosomes on the opposite sides of my cells. They are at the poles. Right? I'm going to draw in my chromosomes. Because not only is that mitotic spindle at opposite ends of the cell, they are now connected to the chromosomes themselves. Right? So the microtubules, they're going to interact with our sister chromatids. And I'm going to draw this out. Let's see. Right, they should be attached on both sides, so each mitotic spindle will have a spindle attached to every chromosome. So both sides of these chromosomes will be attached, one to each spindle. Right, so the chromosomes are attached to the spindles, and the spindles are attached to the centrosomes, which are at those opposite poles. prometaphase. Alright, so nuclear envelope is gone. Mitotic spindle is at the poles, and they are attached to the chromosomes. On each of these slides, I tried to show you the specific blow-up picture of what is happening here, kind of similar to what I am drawing on the board. And now that I'm looking at my slide, I'm realizing I did not explain what a kinetochore is. Alright, a kinetochore is the physical region where the mitotic spindle attaches to the chromosome. Right, so it's on the chromosome itself, it's where the microtubule attaches. Alright, we've gone through prophase. So prophase, we had the nuclear envelope start to disappear, the chromosomes have condensed, and the mitotic spindle started to form. Now in prometaphase, we have the mitotic spindle on either side connected to our chromosomes, and our nuclear envelope is completely gone. There's my picture of the kinetochore. Right, so our kinetochore is going to be close to the centromere. That's that region where the mitotic spindle physically grabs the chromosome. Now we're going to move on to metaphase. Right? I like metaphase because metaphase I think is easy to distinguish apart. It looks very distinct from all the other phases. So again, I'm going to draw my cell. I know my centrosomes are at the poles because that's where they migrated in prometaphase. Now what's happening in metaphase is that these microtubules that are attached to the chromosomes are playing kind of like a game of tug and war, right? They're pulling back and forth until the chromosomes are aligned in the middle of the cell in a row, in a singular row. Right, so I'm going to draw out my chromosomes here.
If they're trying to get it towards the center, I think it'd be more like moving a couch than it would be like tug of war.
It could be keep slightly adjusting.
I feel like tug of war implies that they're like competing and they're like trying to get them both to their side, but if they're trying to get it to the center, it's probably better to describe it more like something like moving a couch where you're pulling back and forth trying to get the couch in that one center spot.
Yeah, it could be whatever helps you remember what is happening here. Right, being the ultimate goal is that our chromosomes are lined up in the middle. Now that middle spot, right, we call the metaphase plate. Now it's not a physical thing. We can't look at a cell and like touch the metaphase plate, right? But the metaphase plate is indicating the middle of the cell. It's the area where these chromosomes are all in a line, right? So they're organized in a single row. I need to finish off my drawing here by drawing those microtubules attached to my chromosomes. Alright, so like I said, metaphase should be an easily recognizable phase because all the chromosomes should be nicely in a row. Alright, let's pause. I want you to review the first 3 stages and tell me what has happened so far. And you can draw this out if this helps you, but I'll give you a couple of minutes.
Basically, chromosomes have already been replicated.
news, you know it. The review. Yeah.
I actually had to craft it, I actually had to do a graph where you had to craft them all out.
Oh, yeah, that's a
I don't think we did chromatic things largely for that one, like we were taught it, but I think for that one specifically, I think my it's 7th grade size, and I think my teacher removed that on purpose. So that'd be easier to look at. But I do remember like actually speaking like, I believe it was a group project too, so it was the idea with this one. I have distinct memories of, I believe making telephase.
Yep, because drawing it out, crafting it out is a great way to study this.
Honestly, it's more of interesting of how many craft products look like pieces of cells.
But like, we laugh too soon. No, I don't.
All right, so far in mitosis, we've gone through the first 3 stages. What is the first stage? Prophase. What is happening in prophase? A bunch of things happening. What is happening to the nuclear envelope? It's dissolving, starting to disappear. What is happening to the chromosomes? They have condensed. And what is happening to the mitotic spindle? They have been formed. They are starting to form. Right? So prophase. What comes after prophase? Prometaphase. What happens in prometaphase? What happens to the nuclear envelope? It's completely gone. What happens to the mitotic spindle? It's completely formed and it's where? It's on opposite sides of the cell at the poles, and the mitotic spindles have attached to the chromosomes. Right? Which leaves our last stage, metaphase. What is happening in metaphase? Yeah, the chromosomes, they get aligned on the metaphase plate. All right, so we're halfway through mitosis. I would say as you're studying mitosis, best way to study, draw it out. Quiz yourself, draw it out, say, start with the cell that has 2 homologous pairs, and do one that has 3 homologous pairs.
Still an easy craft project.
Craft it out.
Lot of craft materials look like pieces of cells.
Yes, whatever helps you study. I think in another class, another professor had the idea of try to make this into a play, right? Act it out. Maybe someone's a mitotic spindle, maybe someone's a chromosome. Whatever helps you remember. All right. I've been noticing on the content checks, a lot of you like it when I have condensed information in one visual on one slide. So I have added that in here. Right? I'm not going to really go over this slide because this is exactly what we talked about. Right? But if you need a visual as you're studying, this slide might be very helpful. All right. So we've gone through prophase, prometaphase, metaphase. We now have to talk about anaphase. All right. So again, I'm going to draw out my cell. My mitotic spindle is still at the poles. I still don't have a nuclear envelope, so I'm not going to draw that in. Right? During anaphase, the chromosomes are actually going to be split. Right? We are splitting apart sister chromatids. Now remember, sister chromatids are identical to one another. Right? So I'm going to draw this out here. Right, so sister chromatids, they were connected at the centromere, right, the middle kind of portion connecting the two together on the chromosome. But now they're going to be split apart. And the mitotic spindle is still attached, and it's the mitotic spindle, the tugging of this mitotic spindle, that has caused them to break apart. So essentially the mitotic spindle is starting to shorten, right? Go back towards the pole. And as the mitotic spindle shortens, it's going to drag along its half of the chromosome with it. All right, so we have split our sister chromatids. Now we have not split our homologous pairs. We still have homologous pairs. Right, so I remember I did homologous pairs by size. So I still have two big and two small going to either side of the cell. So we still have homologous pairs. It's the DNA that was replicated, right? The DNA that was identical to either side of that chromosome, the sister chromatids, that has been split. All right, so we're moving towards the poles. All right, so prophase, prometaphase, metaphase, anaphase. We have our last step, or our last phase, I should say, which is telophase. Okay? So during telophase, let's see. I'm going to draw telophase as one big cell. Right? And I'm going to draw in my halves on either side. During telophase, our nuclear envelope will start to appear. All right, so our sister chromatids that have been broken apart, they have now migrated fully to the poles. They have reached their respective locations on either side of the cell. The nuclear envelope is going to start to reform. All right, so it's starting to reform, so I'm drawing it as a dash here. So essentially what we have is one cell with two nuclei, each nuclei containing exactly half of our genetic information. Our chromosomes at this point are going to start to decondense. Right? So I've still drawn them condensed. If I were to draw telophase, like the later part of telophase, I would start to draw them as, looks like a plate of spaghetti and either nuclei. You want to be able to tell them apart. They are decondensed. All right, now we don't want one cell with two nuclei. Right? We need to split our cell apart. So even though we've gone through the last phase of mitosis, there's still one more step that needs to happen. All right, so we still need to split the cell apart. There we go. And that is done by cytokinesis. We have to split the cytoplasm apart. So I'm going to draw it as two cells next to each other. It's kind of bent in the middle, so it looks like this figure 8 on its side. Right? I've drawn a dashed line showing where the split is happening. Now, where this split is happening is called the cleavage furrow in animals. Right? This is slightly different in animals versus plants. Right? On either side, we have nuclei that have full nucleic membranes, and they have our chromosomes. And I'm going to try to draw them a little bit less condensed here, because they have decondensed down. Right, so at the end of cytokinesis, I've kind of drawn it mid-stage here, right? We should have two separate cells. Right, so again, cytokinesis is the process of splitting that cytoplasm, dividing it equally between our two cells. Right, now like I said, plants and animals, this is slightly different. In animals, we have kind of what I've drawn on the board here. We have this cleavage furrow starting to form, right? It kind of cinches the middle of this very large cell with two nuclei to the point where that one large cell is going to become two distinct cells, right? In plants, we don't have this cinching occurring, right? It's not like we're pulling a drawstring and making it small, right? We actually have this plate that starts to form, right? Because the cells in plant cells, they have that cell wall, that very rigid cell wall that maintains its shape, so it can't physically furrow. So instead what happens is a cell plate starts to form, and we'll eventually divide this cell into two, and that cell plate will become the cell wall. All right. Let's test our knowledge. So I'm going to give us roughly 10 minutes, and I want you to go through two slides here. I want you to go through this table and tell me what are the major events happening in each phase. And then I want you to look at the next slide and tell me which circles represent which phase of the cell based on what you know of mitosis. Right? And I'll give us about 10 minutes. If we finish a little early, that's great, but I don't think we will. I think we'll finish, actually, I take that back. We're going to finish a little early. I'm going to give you about 6-7 minutes here because then I want to go on to the content check, and I'm going to give you one of the answers because we did not make it that far. Yes, I said the magic number.
Yes. That brought in my day.
And counting more mistakes.
Okay about mitosis? That's the process of splitting the cytoplasm. When it finally splits, we would say there's now two daughters. No. Yes. Centrosomes are part of the mitotic. The centrosomes are like those dots at the holes, and the mitotic siblings thing is the cytosome and the microtubules. Jonathan, how's it going? I can tell.
So I got two of the steps.
Okay.
Two is telophase.
Okay. Yeah.
I was trying to comprehend what I was seeing from there.
Yeah.
The thing is that I don't even think that the images do not do a good job of telling what it's supposed to be.
So what would be lined up on a plate? That's a tricky one. Think about the cell cycle as a whole. What I'm looking at is, like you said, it's like a ball. You can't really see distinct from
Oh, yeah, we got to.
Chromosomes are not in it, so it's
Oh, really?
You never know really.
Oh, yeah.
Should take it last. Right now.
Yes, it feels so good. I did it.
I just applied for a job.
Oh, right.
Yep. I think like 90%. Where do you go? Oh, students. This is for this year, right? Where's the house?
No, they went to Jersey Mike's. They didn't have bread, so neither did we have bread. And we were out early 3 hours. I'm not kidding. Did she not know? Did she not see the bedazzled thing? That's really cool. My favorite movies.
You don't have a lab? You're doing the lab on Monday?
Are they gonna bring together? I have hopes for them. Stop.
Alright, I know some of you might still be discussing, but I wanna make sure we get through the answers and talk about that content check here. Actually, content check's not due until Thursday, right? Ah, I don't need to review it. I was thinking it was due today. By the end of class Thursday, you will have all the answers. You should be able to fill out most of it by the end of class today. Alright, well, let's take the last 5 minutes to go over the answers anyways. Alright, my answers are already appearing. So if I start at prophase, what is happening in prophase? Chromosomes condensed. Nuclear envelope is starting to disappear. And the spindle has started to form, yes. Alright. Prometaphase then, what happens to the nuclear envelope? It's gone. What happens to the mitotic spindle? They're at the poles. And have they attached yet to the chromosomes? Yes, they are attached to the chromosomes. Alright, so then they're attached to the chromosomes. In metaphase, what is going on? Yes, all the chromosomes are lined up in a single row along the metaphase plate. Alright, anaphase is our next one. What happens in anaphase? Yes, spindle shortens and they separate, but specifically the sister chromatids separate. Right? So sister chromatids split and fold to the poles. Right? And then telophase and cytokinesis, what is happening here? Nuclear envelope has reformed. It decondenses, the chromosomes start to decondense, and cytokinesis finishes off the process by splitting that cell into two. Right? So at the end of all of these steps, we should have two daughter cells identical to the starting cell. All right. Any questions about these steps? Let's go over our cells in our image here. Let's start with number 1. What stage of mitosis is likely represented? It's prophase. Yes, I would say it's prophase. Because if we look at it, right, there's no particular arrangement, but the chromosomes, we're starting to be able to pick out individual chromosomes. So they're starting to condense. All right, number 2, what would we say? I would say yes, telophase, maybe, and also cytokinesis is happening. Right, now this one is a little tricky. I purposely circled this one, but if you look really close, I think it's just 2 cells on top of each other. But if we see 2 nuclei like that, it should indicate telophase and cytokinesis. All right, number 3. Metaphase. Those chromosomes are condensed. They look like they're all in a row. What about number 4? Anaphase. All right, our chromosomes are starting to split apart here, going to either side of the cell. Number 5. I purposely threw in this one. It's a little tricky.
Cytokinesis?
It's not cytokinesis. It's actually, I would say this is an interphase. It's not yet part of mitosis. This cell, their chromosomes are not condensed. That should be a key indicator. The chromosomes have not condensed yet. It's still hanging out in interphase. What part of interphase? I want to be able to tell you. I don't know if the DNA has been replicated or not yet at this point. All right, and number 6.
Metaphase?
I wouldn't say pro-metaphase, but we're getting close. I'm hearing metaphase. I would accept metaphase. I would also say that, well, I guess I would accept multiple answers here. So maybe I would accept pro-metaphase as it's starting to align at the plate, right? Late pro-metaphase. It could be early metaphase and that hasn't fully aligned yet. It could be metaphase, or late, or I should say late metaphase, and that it might be starting to split apart and almost reach anaphase. This one's hard. It's hard to tell based on this image. All right. Let's see. That is all I had. And again, I put this review slide in there. So homework for Thursday. Keep in mind there's that assignment due tomorrow. I tried to space it out a little bit. It should be a quick and easy assignment. And then content check due on Thursday, and we should get through all the answers.
I think they'll get. I shouldn't be taller than it. I think they are.
Which would make sense for pre-med because those are
Day and a half.
probably listen to that too. Yep.
The adults in my life are making my life more complicated. They're not helping.
The big question is going to be, is my next class going to show up on time?
Right?
No, tell me why.
Well, I already took a psych class, but I think this is
Oh,
more
we
like
don't have
a,
the class, we don't
I don't know
have
if it's
unit
required, I think she's just
testing
saying I
on
should
time.
take it.
Why don't you set the meeting between the two? And then he's been attaching the checks from the
If
third
you
party.
look at
And then
Miss Davis, if you search for like the academic catalog,
No,
Yeah.
the
it
third
should
party.
tell you what's
All the
needed
other
for
Sarah
every
said
new
you
class
wanted
is
this
needed.
meeting.
I think she sent me that.
Okay,
Yeah.
yeah.
Yeah, for section 8 of our lab, which of, I saw in the lab manual that we're supposed to read another one article, that there's supposed to be one article assigned to us. Which
Yes.
one was that?
Oh, great question. I can actually pull it up because I have the slides right here.
Like I recall.
Wait, what content should you do on Thursday?
The one about mitosis and meiosis.
The one on Moodle?
Yes.
Oh,
Yep.
okay.
Yep. Let's see. Are you? That's not your class. Group 1. You are reading sugar and sweeteners. Also, what is diabetes?
Guys, I just have a question for that, just to know if you're reading my mind. I
Yes.
just have to leave early because we have like one-on-one meetings with my coach today. So
Sounds good.
I'll be leaving just a little early.
Well, nothing to turn in at the end of class,
Okay.
so you're good. Yes.
It's like I can just watch like the lecture or the other
Yeah.
when I leave or whatever.
Yeah.
Okay. Thank you.
Corrections?
Yeah.
Perfect. I really need these people when you enter your name on both of them, I'll staple it together in my office. Yeah, I saw that. Another student pointed that out to me. Yep.
It's just that. It's just this.
That, yep. He does. So, me and. First year rule. We're gonna run bumps. I'm gonna have to go back and manually grade it. Yeah. That's actually gonna be kind of a. What? I mean, it's gonna work out in your favor because I tend to get partial points in your favor, whereas Moodle does exactly partial points. So you get like a 0.33 on a question, I can do Yes. Let's see, do I have my files? Since I'm here. My file's like slowly disintegrating on me.
I just asked a random person to open the door. I thought you were talking to someone.
No, I like stared at you and I was like, oh. I'm really curious to see how much the class remembers our new strength.
Why is it been so bad?
This election day schedule kind of messes everything up.
When's our next exam?
Yes, that sounds right.
Oh my, that's really soon.
It's 2 Tuesdays, so in 2 weeks.
This is the hardest year by far.
Really?
See, I've heard different opinions.
I can just.
Guys, I never ate grapes back at home. It only happens grapes per fruit. Why am I so addicted to grapes? I love them.
No, the grapes here are so good. It's half grapes. You just gotta make sure the good ones.
Yeah.
I just like dig around. Yeah, my coach is gonna toss up the mints for grapes. Sorry.
What's that?
Like, yeah, I'm going to Arizona.
Are you going? Wait, I might have missed this at the beginning, but where are you going?
It'd be some girls stay back and some guys stay back. So it's just like the best top 6 people each. Girls and guys take go together.
Oh, so do you like golf?
That's so cool. It's an all golf trip. Yeah, I'm in the top 6 right now.
Like, I'm fairly confident at this
There's
point,
teams
but
behind me. Like all the freshmen girls are in the top 6. Yeah. And there's 2 seniors behind like the bot, like not even
you have any band freshmen, ask for the pile up front. I don't have a stapler, just make sure your name
But I
is
feel like you
long.
would be like the people that are not potential like be in the team like
No, you're good. I see you're avidly doing Oh, it is mine. Okay. Thank you.
I'm sorry. I don't think this girl worked for me. Who did they? I mean, if your back is still really bothering you, one more Sorry, I'm just
All right, let's go ahead and get started here.